COVID in the brain. Autopsies were performed on 12 people with COVID and without COVID. And while the COVID virus was only found in 4 patients within the olfactory bulb, brain, and brainstem, there was neuroinflammation in all of the COVID patients, with previous studies showing brain inflammation and brain scarring in those COVID patients. In this study, they looked at 2 different COVID targets and labeled them with a fluorescent tag so that they could be seen visually, with that bottom figure showing COVID and the brain, and the top figure showing no COVID and the non-infected people, which is why those scores are black, with brain inflammatory cells being seen in both white and gray matter of the brain in those COVID-19 patients. And this neuroinflammation was associated with peroxisome biogenesis. Peroxisomes control cell metabolism, where they break down fatty acids that can harm the brain if they accumulate, and create energy that the brain can use out of this reaction. If peroxisomes are not allowed to do their job, these fatty acids will build up, damaging the protective coating around the nerves, ultimately damaging and destroying them, with this ultimately contributing to things like tachycardia, unstable blood pressure, and brain swelling, by hurting that autonomic nervous system. Back to the study. The COVID virus was found in patients up to 230 days after symptom onset, and the authors discuss how this neurological infection is considered similar to what happens with HIV and is related to other inflammatory diseases like MS and Alzheimer's disease. While quite scary, and while we don't yet know the precise mechanism of how this all works, this peroxisome biogenesis is a good target for drug therapy.